recording.
Okay. So I
And
never presented using Teams, so you're going to have to tell me how to do that.
yep, happy to do that. And we'll cross that bridge in a couple of minutes. So typically what we do on these meetings is we do some general introductions with all the participants on the call, just so you have an idea of kind of backgrounds and things. And then today, Ian is going to be introducing you. And then we'll shift over and make sure you can do your screen sharing and launch into your presentation. And I'm just going to go down the participant list that I see on my screen here, and I'll start off with myself. So my name is Rune Storson. I am a researcher at the Center for Catastrophic Risk Management, currently serving as executive director. And my research interest is safe and reliable critical infrastructures. Sounds straightforward, but actually turns out to be a lot there, which is interesting. So next, let's go to Emery.
Hi, my name's Emery Rowe, and I do or have done and continue to do research on high reliability management, particularly of complex infrastructures and complex systems. Thank you.
Cool. Esfandiar?
Okay, hi, good morning, everyone. Yeah, Esfandiar Imani. My expertise or experience been past 25 plus years in energy, renewables, and last 10 years in risk management, actually.
We should mention that we started a new nonprofit, Safer Cubed, for safe, reliable, resilient, and risk reduction. So hopefully the IRS is going to sign our papers in the next month or so, and we can actually start doing some good in the world.
Nice.
All right, so Gianpaolo from Italy.
Yeah, hi everyone. I'm Gianpaolo from Italy. I'm a civil engineer, graduated at the Polytechnic of Milan. I worked for Tesla till last year in the supercharging department. So I work in the electric vehicle environment. And actually, I'm attending an MBA program in the Bay Area at Cal State University.
Cool, thank you. Heather, do we have Heather on the line? Heather looks to be muted. Let's see if I can unmute you. Oh, she's got a noisy background. Okay, we'll skip you, Heather. Ian.
Ian Mitroff. I work in crisis management and complex, messy systems. And let me say a brief word about Leah.
It's not time yet, Ian.
Not
We're
time.
going to do the introductions and then you'll introduce Leah. Yeah.
Okay.
But I like the enthusiasm.
Great.
Okay, Kathy.
Hi, I'm Kathy Schaefer. I'm doing research at the University of Davis, UC California, Davis. Research is in flood risk, but also in issues of community risk in general. So thanks for letting me join.
Cool. Peter Martelli, are you on? I don't see an icon for you, but I know that we were chatting. No. All right, so let's see. Before we start, let me just give a quick overview on the Center for Catastrophic Risk Management. So CCRM was founded in 2005 in the wake of Hurricane Katrina. We're a global group of applied researchers from academia, government, and industry who've come together to develop pragmatic solutions for the complex, multi-jurisdictional, messy problems of the 21st century. We conduct fundamental research on catastrophic risk through empirical data by identifying and engaging targeted teams whose expertise are uniquely tailored to each challenge. CCR aims to help make the intractable manageable. So that's a little bit about us. And now I will pass the baton over to Ian to introduce our speaker, and then we'll see if we can get the screen share to work.
Just very, very briefly, that I came across Leah by reading a book by the person who runs the center of which he's a part, A Crack in Creation, talking about this very powerful and yet frightening technology, gene editing. And I had lunch a few times with Leah, and she has a very important job as one of the policy people in the center of which she's a participant. And her so-called boss, Jennifer Duda, just won the Nobel Prize in Chemistry this year. So without further ado, let us hear from Leah. Thank you, Leah, for coming. And we've tried to get you for so long, but given COVID, it's been a real journey. Thank you again.
Yeah, pleasure to talk to you all. Shall I attempt to share my screen?
Yes, let's do the attempt.
So you'll all have to bear with me a little bit because I've only used Teams once before and I've never shared my screen. So we will see how this goes. Okay, I see something that looks like I should be sharing.
Yeah, there should be like a rectangular thing with an up arrow.
Yes.
Why Microsoft can't make it easy like Zoom is beyond me, but here we are.
I see it. Okay, here we go. Can you see like a title slide?
Yes, yes.
Okay.
CRISPR and the power to rewrite life's code.
Yes. Okay, perfect. Oh, it's on auto. Hold on. Sorry. I don't know. Can you? Oh, my screen just went blank. Did I lose you all?
No, we see your desktop.
You see my desktop.
Now I see myself in your video feed.
Oh.
And there's Peter.
Okay, hold on. Let me. Okay, let me see what is happening here. Sorry about that.
Okay, that's why we're here talking about technology and its problems.
Yeah. We always joke, we have these Zoom rooms in the building, and we often joke about how many of us it takes to get them to work, and yet we're working on these very highly technical problems. I'm not sure why this isn't.
And it's not possible to move the bar down to the bottom of the screen as it's in the middle and it interrupts the presentation. I guess
Yeah.
not.
So if you want, you can email me the presentation. I can share my screen. That's
I think
backup
I
we need it.
solved the problem. Hold on, let me try this again.
We're all about workarounds.
It's thinking. Okay.
Part of our self-resiliency.
Ah, very good. Okay, we have the button here. Screen 2. Can you still see the intro slide?
Yep.
Yep, see it?
Oh, and I can see you now. Good. Okay, that's better. Okay, so hi. Thank you for having me. Yeah, so, you know, this has been, I guess, a long time coming, this talk, and so I'm happy to be here and share an overview. It's hard, when I was thinking about what to present here, it's hard to pick an issue. So I've kind of opted for a array of kind of cursory level introduction to some of the potential problems and risks that we are seeing in the CRISPR field right now. So here we go. So is it Ian?
Yes, Ian, yeah.
I've always thought it was Ian, my bad. I'm sorry, I apologize. So as Ian mentioned, I work for the Innovative Genomics Institute, which was founded by Jennifer Doudna. And we are a joint institute between UC Berkeley and UCSF. We are a nonprofit, and we have this vision and this mission. And so we work across application areas, trying to bring together different expertise to apply genome engineering for the benefit of humanity and in hopes that applications can be accessible and ethically and equitably distributed. So our main program areas are shown here. We have 4 program areas. We have a program in human health, climate and sustainable agriculture, advancing the technology itself. And then I work in the public impact program, which is meant to be connecting with each of these application areas and to help both inform the applications, but also to communicate between the general public, important stakeholders, and policymakers. So we have a fairly broad charge. And we think that it's fairly unique that we have this structure. I haven't really seen a predominantly scientific institution have a program like this integrated directly with the others. And this was really Jennifer's inspiration. So as I think you maybe gleaned from reading her book, she really has been an advocate for considering not just the scientific and technological issues, but the societal and philosophical issues that are raised by this technology, which I'll explain how it works in a minute. So when she founded the Institute, she founded this public impact program to be an integral part of it. So we see our mission in the program to align genome editing applications with societal needs and values. So both internally, how we do our work at the Institute, what we apply it to, as well as be part of global conversations about its governance and its use. So here's kind of the structure of how I'm decided to organize this talk. I'm going to tell you about the technology itself, so how it works, where it came from, so that you kind of have a deeper understanding of what the potential possibilities are with it. And then I'm going to go over some of the applications and some risks and how sort of I've been thinking about the different risks. And then lastly, I'll touch on a few of the most controversial applications and sort of what's controversial about them and what sort of risks they present as well. And at that point, we'll have questions. So let me dive in. So we'll start with how it works. I'm not sure how many people have had recent course in genetics and genome engineering, so I figured I would start kind of bringing everybody onto the same page. So a few terms, I like to use this analogy to a book. So if you think of a genome as being a book of instructions, and the entire book, maybe even volumes of different books, contain all the instructions to make a physical living being, whether that is a single-celled organism or a human. All living things have genomes, and those genomes are organized, like books are, into chapters. And so you can think of a chromosome, which is an organizing unit of a genome, very similar to how a chapter is an organizing unit of a book. And then within those chromosomes, we have genes. And genes you can think of analogous to sentences in a book. So each gene is a contained, discrete sentence, so to speak, about the instructions of how to make an individual or an organism. It takes many sentences to construct an organism, and often, features of a person, for example, height is dependent on hundreds of genes, but there are some things that are dependent on one gene. And so there's a variety of ways that these genes can be organized to give these instructions, and some of them are redundant and some of them are not. And also very similar to a sentence, genes have architecture. So they have a beginning, a middle, and an end, same as a sentence. So we can recognize a sentence because we see that it starts with a capital letter and it ends with a period. So genes are very similar. They have a certain characteristic at the beginning of them that we know indicates this is the beginning of a gene. And they have a characteristic at the end that tells us the same thing. And they're written in a language very similar to sentences, written in letters. So in the case of genes, we have chemical bases. So they're depicted here as A, C, T, and G, and those are just the first letters of the chemicals that are part of the bases. And it is a code. It's very simple. every 3 letters put together makes one codon, and one codon is part of a protein. So you build these proteins by reading the code, and those proteins are the things that make up your body, and they do work. They'll make lipids, they'll metabolize certain products to make other things that are not proteins that are still part of your body. So many aspects of every being is encoded in your genome. So what is CRISPR? So knowing this, knowing what genes are and what genome is, we have to go back over 30 years ago to the beginning of the story of this discovery of CRISPR. And it started kind of right around when we were getting decently good at understanding what DNA is. So back in the late 80s, we were starting to be able to do sequencing. And so scientists were sequencing all kinds of different things, learning about the DNA in them. And they sequenced a bacteria. And they found inside of this bacteria, and this was a streptococcus, I believe, inside this bacteria, they found a very strange pattern. So you remember I told you that genes have a very characteristic architecture, like a sentence. They have a beginning, a middle, and an end. So they found these genes that have the beginning, middle, and an end to them, but then they also found this other region of DNA upstream of it that didn't have any of those characteristics of a gene. They had a very unusual characteristic, which was that they had no start or end, no period, no capital, but they had these repeating small segments over and over, sometimes hundreds of times, interspaced by these random chunks of DNA that were not the same. So you had hundreds of random bits of DNA interspersed by these consistent repeat bits. So that looks not like a gene at all. And it was about 20 years until scientists figured out what that was. And what it is CRISPR. So I think it's kind of useful to understand what CRISPR stands for. So it stands for clustered, regularly interspaced, short palindromic repeats. So now that you understand the array, so they're clustered, they're all together, they're regularly interspaced by these repeats, and they're short palindromic repeats. So they happen to be the same at the beginning and the end. So that's what CRISPR is. It's just referring to this weird region of a bacterial genome that was discovered in the late 80s. And, oh, there we go, sorry. So about 20 years after its discovery, scientists started to get a clue as to what this region actually did. And the clue turned out to point to viruses. So bacteria are living things, and turns out most, if not all, living things can be infected by viruses. And so this is actually a real electron micrograph image of viruses in pink attaching to a bacterial cell. And when they inject their DNA into the cell, they hijack the cell's DNA reading and writing machinery, and they make so much of more of their own tiny little viruses that they explode the cell. So here's one kind of at the verge of making too much to explode it. So it turned out that those odd random spacers, is what they called them, in between the repeats, could be mapped on to known bits of viral DNA. And so this really was identified because of increases in our ability to do DNA sequencing. So we were generating more and more sequences, and they were able to match these strange CRISPR array spacers to viruses, and lots of different kinds of viruses, all of which were known to infect that type of bacteria. So it turns out that the way it works is when a virus infects that bacteria, the bacteria makes a copy of that tiny piece of DNA that's unique to the virus, and it inserts the copy into this region of its own genome, and it interspaces it by these little repeats. And every new infection, if the cell doesn't die, it memorizes which virus it was infected by including a new chunk of the virus into its own genome. So every one of these is a memory of a past infection that the ancestors of that individual cell had survived. So if it survives, it's able to create a memory of that infection, and then upon a future infection by that same type of virus, it now has the ability to recognize it like an immune system and attack it. So it's constantly creating short little copies of each one of these little memories of viral infections into what's called a guide RNA. And RNA is very similar to DNA. It's just a shorter, more transportable version of DNA, basically. And so the cell makes these little transportable copies of the spacers called guide RNAs, and they get loaded into this enzyme called a Cas cleavage complex. And this enzyme is just constantly scanning the cell, looking for invaders that match its mugshot, which is the guide RNA. So it's constantly trying to match the guide RNA to DNA. And in the event of a subsequent infection by the same virus that the descendants of this cell had seen before, it matches, and then it cuts the DNA. And cutting that DNA kills the virus. It stops the ability to infect, because the virus needs to be able to have all of its DNA intact to make more and more viruses. So CRISPR is a bacterial immune system. And a lot of this was known, except this mechanism, how this happens, was unknown in 2012. And the thing that Jennifer won the Nobel Prize for was not all the stuff that I just told you leading up to this. It was the part about how this works and what you can do with it. So Jennifer and her collaborator, Emmanuelle Charpentier, did some biochemistry experiments to see, we know there's RNA, we know there's this protein, and we know at some point this gets cut. How does that happen? Well, they showed that it required this RNA, and that in fact, the RNA is an exact match, letter by letter, to the piece of DNA. And they showed how it works, that it binds and it cuts. And that was transformational, because what it meant was that now we as scientists have an ability that if we know the DNA sequence of any gene anywhere, right, we can make a little tiny copy of that sequence that we want to target. We can load it into this protein, and we can release that protein to do its work, and it'll find it and cut it. And so it is a programmable DNA cutting machine. And she was able to show that she could repurpose it outside of the bacterial cell. So she took just this part, took it out of the cell into a test tube, and was able to show that she could make this whatever she wanted, and she could give it any kind of DNA and it would do the same thing. So that was the eureka moment of, we've discovered something that we can use, that we can program to find any DNA and cut it. So how do you apply that? Well, so genome editing, as I just mentioned, it's a cutting mechanism. So people often say, oh, we can rewrite things. Yes, we can, but the rewriting is more than just the Cas enzyme. So the enzyme itself, if you know your gene, you make a little copy of it here, you give this RNA to this protein, which is shown here. It'll scan around a genome, try and find a match. And if it finds a match, it opens up the DNA and it cuts. But then now all you have is a cut piece of DNA. So genome editing really begins with the cut, but what happens after that is really the critical part to being able to use it. So there's two general ways that you can use that cut to make a change in the genome. Turns out that our cells are constantly bombarded with chemical and radiation mutagens that are constantly cutting and damaging our DNA. And so we, along with many other living things, have evolved natural mechanisms to deal with that. And so we have these complexes in our cells that are looking for damage to our DNA and repairing it. So what we do as scientists is we take advantage of that natural repair mechanism in our cells. And there's two. One is called non-homologous end joining. So that's when your cell sees a break and it just puts the ends back together. It really literally makes a new chemical bond between one strand and the other strand. And then the other mechanism is called homology directed repair. And this is very useful because we happen to have 2 copies of every gene. Some organisms like plants have multiple copies of every gene. One, for us, it's one from our mother and one from our father. And so if you have one of those copies is broken, you have another copy already as a backup. And so the cell has a mechanism where if it can't just put it back together end to end, if there's like a missing chunk or a problem with it, it can find what's called a homologous pair, meaning something that matches it exactly. And it uses it as a template to fill in the missing chunk. So that's called homology directed repair. So we scientists have taken advantage of those 2 natural pathways by either using non-homologous end joining to create a mutation or using homologous directed repair to make a new known mutation or to insert a new chunk of DNA or to take away a chunk of DNA. And so the reason that this works is because in the natural setting of your cells, when it tries to just put the damage back together and re-anneal the 2 strands, once that that joining happens, there's no longer a cut, a new cutting mechanism. So it's joined and the repair is over. But with genome editing, you have supplied this machine that will continue to cut at that same spot as long as it matches the RNA. So if there's no mutation at that target site, then it'll just keep cutting it until eventually there will be an accident in the cell's ability to put the 2 back together. It might lose a base pair or make a little bit of a mistake, and then that's no longer a substrate for the machine, and you end up with a mutation. And these can be small deletions. They can be, a single base pair switch. So instead of an A, you get a T. And then homology directed repair, we figured out that instead of letting the cell use its natural homologous partner, we can supply it with a really short donor template that matches it, that has the homologous pieces on it to each side of the DNA that we cut, and also has whatever we want in the middle. And if we just put enough of this, then the cell will use this instead of the homologous natural partner. And so we can, in that way, make either one single known directed base pair change, or we can make large insertions. So that's a lot of technical stuff, but hopefully that kind of allows you to think, well, what sorts of things could this be used for? And it turns out a lot. So, not a long time ago, actually, maybe 10 years ago, it used to be that in order to do experiments on genetics, you needed model organisms. So you needed an ability to change a gene to see what a gene does. And there were only so many organisms that were tractable to altering their DNA. And so this shows essentially the extent of the model organisms that we had for decades prior, only 10 years ago. So now with genome editing, it turns out, perhaps to some people's surprise, that it is extremely effective and it can be used in any kind of cell that anybody's tried it in so far. So we went from a handful of model organisms to over 300. We've tracked for a while. We've stopped tracking because there's so many, but we know there's at least over 300 organisms so far that have been edited with CRISPR. So why would you want to do that? Well, one reason is certainly for experiments. So this has already completely transformed the way we do biology in the lab in terms of just fundamental understanding of functions of genes. So you can do experiments that you never used to be able to do before. And it's allowed us to ask questions and learn answers about basic processes of human cells, of plant cells, of all sorts of things. But there's also, you know, practical applications of this. And that's because of the function of the genes themselves and the fact that they, you know, sometimes even a single base pair change can have a very large effect on the organism. So for example, if this gene at this position had an A and we changed it to a T, you know, the analogy with the sentence would be, maybe it used to say twinkle, twinkle, little star, but now it says twinkle, twinkle, big star. It turns out that this exact T to A mutation is known in human health to cause sickle cell disease. So sickle cell, I don't know how many of you have heard of it, but it's a blood disorder. It creates sickling in the blood cells that can then get clogged in arteries and veins and cause loss of oxygen to organs, to tissues, et cetera. It can be lethal. In first world countries, it's often not lethal until 40s, but in third world countries, you know, newborns and young children often die from it. And this is caused by a single letter change. So this is a very, you know, attractive target for being able to create a therapy by fixing this mutation in the genes. And in fact, this is something that we are doing at the Innovative Genomics Institute and others are doing. You may have seen in the news, the first CRISPR edited sickle cell patient was just, I think, 2 years ago. And she has seen absolutely no effects of sickle cell. So she seems to be fully cured, which is great. And the idea here is that because these are blood cells, you can simply take blood cells out of a patient. You can try and extract what's called stem cells. These are cells that are sort of like young cells and they make new cells. So they make the blood cells. You isolate those, you add in your Cas9, you add in the guide RNA that matches that mutation in the blood cell, and it makes the correction. And then for us, the way that we're doing it is we give it a protein and a short piece of DNA template to correct that mutation. And this is all done, we extract the cells, and then this is done in a hood by a person in the lab. The machinery, the CRISPR machinery, then degrades after it's made the correction. And so now you have no trace of the foreign material. All you have is the corrected gene. And we put those back into the patient. And when they get back into the patient, they are then able to start producing new blood cells, and they will do it without the sickle mutation. So this is super exciting. There's lots of heritable diseases that are genetic, where we know there's a known causal relationship between a mutation and a very severe genetic disease. So sickle cell is one. It's one of the early targets, but there are others. People are interested in certain eye disorders. There's looking at developed or inherited deafness, other things like cystic fibrosis, etc. So that's human disease. But again, if it has DNA, we can edit it. So we can also use it for agriculture. So this is sort of, some people would call this GMO 2.0. We can talk about the use of that terminology and what risk that involves. But basically anything that you used to be able to do with the old GMO technology, you can now do with genome editing, but better, more precisely, without that foreign DNA. And you can do things that you never used to be able to do. Like you can make deletions, you can add just a single base pair change instead of adding an entirely foreign piece of DNA. So there are ways that we can now tune agriculture very precisely that we could never do before. So this has applications like for climate change adaptation, so making crops that can survive better in drought or in flooding. So For example, one of our collaborators in Davis works on rice tolerance, flood tolerant rice. So she's making rice that's able to handle, the increase in flooding and the length of flooding periods, and still be able to produce rice for farmers mostly in Southeast Asia. There's also risk of pests and pathogens decimating crops, and if you can introduce a variant into a gene that allows the crop to resist pests and pathogens, then you don't have to use pesticides. So that has a lot of appeal. And there's also potential for livestock applications. There's a person in Davis that's doing this as well, who is trying to make more humane mutations in cattle. And I'll tell you about that in a second. So, but the idea being that, we can really use this in anything. So what are the risks? Those are some of the kind of potentially promising and exciting applications. There are risks with certain other applications, but also with those. So, anytime there's an advance in science, it poses a question that doesn't have scientific answers. So, we've created this new technology, so now we know we can change the DNA of any living cell. Should we? What should our relationship with nature look like? What are the unintended consequences that we haven't really thought through? Are there known unintended consequences that we can do something about? There's questions about benefit and risk and who bears those. So who does an application benefit? Who does it harm? How can we responsibly use this technology? And what does responsible governance look like? So I often like to break these risks down into sort of these three categories, and they're overlapping. I probably should have shown this more as a Venn diagram, but I think often when people talk about risks from genome editing, maybe when scientists talk about risks from genome editing, they're really talking about the technical risk. They're thinking about, well, what if it doesn't make the right edit? Or what if, I use a gene to encode the editing machinery, and then that machinery is able to edit some other organism that I didn't want it to. Those are technical risks. So those are because of the way the machinery works. And so the example that I just gave is an off-target effect. So what this means is, if you remember, I mentioned that if you know the sequence of the gene that you want to target, or the piece of DNA that you want to target, you can type that into a computer, make a copy of that section, produce it as a little RNA, and give that RNA to the Cas9, and that will be your guide to find the match. Well, it's very accurate, but sometimes, a very small percent of the time, there will be really similar sequences somewhere else in the genome that maybe have one difference in base pairs. And most of the time, this is so accurate that it won't cut that. But every once in a while, it will. And so what happens if it cuts somewhere where you didn't want it to cut? And those are called off-target effects. And there's multiple types of things that could happen there. Often nothing, it'll cut an off target and there's zero consequences. Other things that could happen would be rearrangements of the genome. So if there's multiple off targets and it cuts in multiple places, those could now accidentally rearrange. Instead of reannealing back together the right way, they could reanneal to a different thing. Or if you had, other foreign DNA in there, that could all of a sudden get incorporated. So those are technical risks. The good thing is that most of those have been dealt with at this point. That's been one of the biggest concerns from the scientific community, and has received some of the most, I would say, R&D in terms of the use of CRISPR. So we now have methods for doing editing in human cells, for example, where we have zero detectable off-target cuts. That's not always the case, and it depends on the way that you're using it, the type of cell you're using it. So it's still something to keep in mind, but it is actually not as big of a risk as maybe people once thought. Another reason being that I think what a lot of the public and other people don't realize is that cuts in your DNA are very common. So the whole reason, if you remember, that we'll find it here, the CRISPR makes the cut, and the reason that we can use this is because of the natural processes in your cell to repair the cut. And all living cells have some kind of mechanism to repair and fix broken DNA because it is a very common occurrence in cells. So that's something that often gets missed in the media, is that yes, you might have a very small percent of the time an off-target cut, but actually a much larger percent of the time your cells are getting cut by other things like UV rays or chemical mutagens in your environment, and they repair it and they're fine. So we've kind of, for the most part, dealt with some of these off-target effects for most of the applications. There's other things still that we're looking into, like immune response for using it as therapies. These molecules come from bacteria. So it's possible that human cells might view this as a foreign object and attack it. That would be very bad for the patient. So that's still kind of an area of potential unknown risk that's being researched at the moment. Other questions of persistence, so both in therapeutics, but also in the environment. So let's say you make an edit in a crop, but you do it in a way that encodes the gene for the editing machinery. So that gene will then persist in that plant, and can it be transferred to another plant? If it did, would it matter? Would that other species even have a matching sequence for it to cut? Some of that is still a little bit unknown. It presents sort of a long shot risk, but it's something to mention. So those are examples of technical risks, I think, that get most of the attention from scientists. I think from the public, there's kind of 2 and 3 tend to be more in the view of the public, whether they realize that they would be categorized this way or not. But there's this concept of philosophical or moral risk. And so an example of this is if we think about therapeutics using CRISPR. So I gave you the example of sickle cell. Sickle cell is a debilitating disease with a causal relationship to a single point mutation. So that would be something using it for treatment purposes. There are other things like, for example, we know a lot about genes that correlate with strength and with muscle twitch. And actually some athletes take gene supplements. They're not allowed to, it's forbidden, but it's been documented to increase their strength or increase their speed of their muscle twitch. If we could encode that, then you could create a stronger, faster person, right? So this would be enhancement. You're not solving a debilitating problem, you're making somebody enhanced. But then there's this kind of in-between space, or maybe even that's a separate thing, which is something called prevention. And I think this is really where kind of the philosophical and the moral risk comes from. And so this is, as we learn more and more about genomics, we learn all kinds of ways that multiple genes work together to create certain traits or certain phenotypes. And we can learn a lot about correlation. And so what I'm showing you here are polygenic risk scores. So, somebody that has diabetes, for example, there is a genetic component to that. There's a correlation. So we would see, people that have this type of mutation down here maybe have a low risk of ever getting diabetes. People that have it way up here have a high risk. So that presents 2 philosophical questions. One is, should there be is that something to treat or not? Diabetes is also a social disease in the sense that it is very even more highly correlated with diet and environment than with your genes. So are genes the right way to treat it? Or should we be fixing kind of the underlying societal causes of diabetes? The other is that if you were to make a mutation in somebody's cells, these are not causal, they're correlative. So there's a chance that even if the person had, this high risk mutation, they may never develop diabetes. So that is very distinct from something like sickle cell, which is a causal mutation. So that's an example of kind of philosophical and moral risks. There's other kinds of social risks, like what is a disease in the first place? So an example of this question is with the deaf community. And we at IGI encountered this fairly early on because one of our investigators was interested in genes that cause deafness. And we learned very quickly that there is an entire culture and language and community around deafness that do not see themselves as having a disease. And so simply the idea of fixing deafness is extremely threatening to them because it threatens their culture. If you were to eliminate deafness, you would eliminate an entire culture. So how do you deal with that? Another example is actually sickle cell. So often things that seem like obvious positive uses of genome editing can have risks to them as well that are social risks. So sickle cell disease is a disease that affects predominantly people from African descent. And in the US, it's African Americans that make up 90% of people with sickle cell disease. And what this has meant historically is that it has been an underserved disease. It turns out it's actually the very first disease ever discovered by scientists to be a molecular disease. So it is the definition of molecular disease. It was discovered and defined it. We've known about it the longest, and yet it took until 2017 to come up with only the second treatment for it that was FDA approved. So this is very clearly tied to race and to social constructs and social ills. So what this also means is that on one hand, it is super exciting that maybe the first beneficiaries of this new technology could be the same groups of people that have not benefited from scientific advances in the past. So that's super promising. But it also runs the risk of if something were to go wrong in these early trials, now you have the Black population who is bearing the risk of this brand new untested technology for everybody else. And so how do we do that in a way that provides health justice and doesn't sort of extort the Black community as being kind of the first to be experimented on, knowing the history of experimentation on Black people in the US and around the world. So that is a risk, and that's a social risk. The other social risk to mention with therapies is equity and access stemming from cost. So gene therapies are extremely expensive. The most expensive one is from Novartis. It's over $2 million. So how is anybody going to pay for that? If we do enough of these, even if they're covered by insurance companies, you will bankrupt the entire healthcare system if we do enough. So this is starting to look like it could be a really exciting therapy for very rich people. So how do we make sure that this doesn't exacerbate health inequities here in the US and around the world? And let's see, I'm coming up at the end of my time. So let me give you, I'm gonna jump because I don't wanna take up too long. Let me give you another example here, which is in agriculture. So I mentioned the GMO connection. So types of plant modifications. People have been modifying the DNA of plants for millennia. traditional breeding is modifying DNA. It is a crude, so to speak, way of doing it because you don't really know what genes are being exchanged or what kind of off-target, so to speak, impacts those might have. But this is something that we've been doing for thousands of years as a species. So I have here a table that tries to kind of highlight the differences between types of breeding. So traditional breeding is just your regular crosses. You get the desired gene when you do it this way, but you also get a bunch of other stuff that you don't know what it is, and it has to be bred out, so it can take a very long time. It's also limited by species barriers. So if you want a trait and you can't find a variant of that crop that has that trait, then you're out of luck, because you need to be able to breed it with an existing species that can breed together. So you can't cross two different species. Mutation breeding was first started in the early 1900s, in 1928. And this is using radiation or chemicals to randomly mutate DNA. So again, as I mentioned, our cells are constantly bombarded with mutagens. This approach is to bombard them even more with mutagens and get sort of in a random array of mutagenesis. And then you put those mutated cells under selection for the kind of features that you're looking for. And eventually, if you do enough of these rounds, you'll start to select out a characteristic that you want. And then there's transgenic breeding, which is what most people know as GMOs. So this is where you take a gene and you put it into a crop or a species. This was first started in the 80s. The advantage is that instead of having random mutations along with the one that you want, or having a bunch of other genes that come along with the one you want, you're only inserting the one that you want and not other stuff. And it's not random. You don't have all this random mutation. You do have random insertion of that gene, but you can also overcome species barriers. So now with genome editing, the advantage is that you can do what you did here, except you can do it without random insertion. You're not limited to a whole chunk of a gene. You can actually just make a single base pair change if you want. And there's no species barriers, and there can be you can do it without any foreign DNA whatsoever. So the end result looks at the genetic level identical to what you could get with traditional breeding. So this has spurred a whole conversation of how is it going to be regulated around the world. In Europe, Europe is extremely averse to GMOs from a societal level. So they have a different way of regulating than the US does. They decided to enact regulations that are based on the technology that you use rather than the product that you produce. The US is interested in the product that you produce, and it's somewhat agnostic to the technology that you use. So there's already been this kind of split in approaches to regulating GMOs. And now we add on top of that this new technology where it's not clear, should it fit under a GMO category or should it not? Because it actually, you could make things that don't look anything like a GMO. They look like a natural product. And so Europe has decided, no, we're going to regulate them as GMOs. And the US decided, actually, there are going to be exceptions that we give. Where if you're making something that truly, in the end, the product could have been developed by traditional methods, then we won't regulate it at all. And what this has led to is potential public perception issues. So scientifically, it is a very robust interpretation of a way to regulate. If we don't regulate traditional breeding and you're making something that looks exactly like a product from traditional breeding, then it shouldn't be regulated. That is scientifically sound, but it presents a public risk in terms of perception. And so what we saw in even the scientific press is here, the first gene-edited mushroom escapes regulation. Here come the unregulated GMOs. So it's already being framed as like, gene editing is somehow even potentially worse than GMOs, because now we're trying to evade it and we're coming up with sneaky ways of getting around regulation, when in fact, we didn't escape it. It was determined that it's not necessary because we produce something that looks exactly identical to a natural product. But this is a real risk, and part of that is because there is such a large aversion to GMOs. If you compare, scientists to U.S. adults, most AAAS member scientists think that eating GMOs is safe, whereas most U.S. adults think it's not safe. So there's a disconnect in terms of the science, but what is often missed is that what many people are talking about when they're concerned, they point to, maybe it's not safe, but if you dig in a little bit deeper, it's often other things that they're concerned about, which are not scientific issues, and they're real issues that need to be dealt for genome editing as well, which are who is profiting. So with GMOs, we saw Monsanto come out as this giant monster of a company that got all this money and controlled all of the GMO products in the world. So people have a problem with that. Who is benefiting? Is this really helping small farmers, or is it really just helping Monsanto make money? And then there's this concern about what is natural in the first place. And that's a values question. It's not a scientific question. And so, genome editing communities are out there saying, no, genome editing is safer. It's even better than GMOs because it's more precise. It's not random. We can really control it. You should trust us. And then this happened. So I mentioned in Davis, there's a researcher that's working on cattle. So she has edited this cow that you see here, which is a dairy cow. And the reason is that dairy cows have horns. You never see them because when they're born, farmers will burn the horn buds off in their heads, and it's extremely painful and it's horrific. And they do that because if they're in close quarters and if the farmer's in there milking them, it can be very dangerous with horns. So they put these young calves through this really horrific procedure to remove horns. So she thought, well, there are natural versions, not dairy cows, but other cows that don't have horns. What if we took the genes from those cows and moved them into the dairy cow to make a cow that just naturally doesn't have horns? So she did this. Sure enough, she produced these dairy cows that don't have horns. They became sort of the poster child of the humanitarian good that genome editing can do and how people need to get out of the way and let this happen. It was not allowed on the US market because it's being regulated as a drug. We can talk about that if you want. But then it turned out after a few years of being out and having conversations with actually the Brazilian government to introduce an entire herd of these cows, it turned out that there was a mistake. There was an off-target cut. They did not screen it appropriately. They didn't do the right controls, and they missed something, which was there's an antibacterial gene that you use when you're cloning, when you're making these DNA plasmids that they use, and that got inserted into the cow. So it probably won't cause any harm to anybody, but it creates a public perception problem and a real issue if people are using this technology and they're not doing appropriate controls, they're not doing appropriate screening, and they're out in the world saying, it's so much better than GMOs and so precise and trust us. And then this happens. So maybe I'll stop there because I'm taking too long, but just to give you an overview, there's also, of course, the very obvious controversial topics of heritable genome editing. So editing germline cells, embryos, or sperms and eggs. So being able to change an entire generation. There's also genome editing for conservation. So editing environments with the intent of either combating climate change or conserving species. And then there's a variation on this, which is called gene drives. And this is basically taking the genome editing technology and making it self-propagating. So normally you would use it, it would cut, it would be done. But if you can add it in this certain way called a gene drive, you can then have that ability to cut become heritable. So when a mosquito, for example, gets a gene drive and it mates with a wild mosquito, it will pass that cutting mechanism on. And it will do it in a way where every one of its offspring has the cutting mechanism, and it will cut the gene that it's meant to. So this has been proposed for things like combating malaria, so making mosquitoes die, eliminating mosquitoes altogether, or providing mosquitoes with a certain trait that allows them to be less likely to carry the microbe that causes malaria. It's also been proposed for conservation purposes to, for example, get rid of invasive species on islands. So you can see it can sound scary. It also has a ton of potential good. If we can use this to combat climate change, you know, we also have to ask, there are risks in acting, but there are risks of not acting, and how do we weigh those? Often when people are worried about this, they think about, well, what if something goes wrong? What if something doesn't get used and we continue to see the devastating effects of climate change? We lose species biodiversity. There are balances to be had between risk of acting and risk of not acting. And are there ways that we can mitigate the risks of acting to balance that, to be able to actually apply this for good? So I will end there.
Okay, I have a question, if I may. Leah, can you hear me?
Yeah, let me turn off my share so I can see.
Okay. Leah, the question I have is, given the social philosophical risks, what, if anything, do you wish that the center had done earlier to foresee the risks, to mitigate? It's like saying, what are the chief lessons that you've learned and you wish that you had put into force earlier on? One other thing I would ask, I've heard that there are now CRISPR kits that almost anybody can get at home. It's like grow your own organism, which is frightening beyond belief. Anyway, going back to the first question, what have you learned that you wish that you had foreseen and that you had acted earlier to help mitigate these risks?
Sure. I have to think about that a little bit. So the reason that it's hard for me to answer immediately is because I think we have to maybe separate between the field and IGI as an institute. And so this is something that I also struggle with in my job, which is, you know, we are trying to be as responsible as we can. We're trying to participate in these global discussions of the ethical dilemmas that are being presented, and as well as the biosafety risks that they present. But there's a lot that's happening that is the field, right? And there's only so much that you have in your control. And so the thing that I think was done well, actually, was very early on in terms of heritable editing. Jennifer was able to recognize, even before it was used in this way, that there was a risk that it could be used for editing embryos and designing babies, right? And so she calls a meeting of people that she trusted to talk about, well, what do we do about this? And that led to basically, and I'll add the other thing that I think she did right there was not try to answer that herself, or not presume that she knows the definitive answer, but to rather say, this is an ethical dilemma that is coming down the pike, and we need to do something about it now. And she kind of put out the call for scientists and social scientists, ethicists to all come together and start talking about this. It turned out that it was being used in that way. And just 2 years ago, we had the first babies born that were edited. I didn't talk about that, but it was horrible, the way that it was used. So violated everything that all of the kind of experts were talking about, the ways that you shouldn't do it. There was a moratorium being called for. There was general consensus almost everywhere in the field that it shouldn't be used, that it's not safe right now for that. And yet it was. So I don't know what you do about that, because that was, I would say she approached that correctly. And yet we still ended up with a case where it was abused. Other things that I think are maybe more tractable is the example with the cattle that I told you about. So in retrospect, I would have loved to see the editing community come together and make sort of a best practices for evaluating your edit. And make sure that people that are using the technology for products are evaluating the effectiveness, the safety of those products correctly. Because they didn't. They didn't even look at the entire genome sequencing, which is shocking to me. So in retrospect, we should have done that as a community.
Just one quick.
I am willing to do that right now for therapy.
Should there be an ethicist in every one of these teams from the beginning?
It's a great question. I mean, I would say yes. Actually, I would maybe change it. I think the trouble with having an ethicist, and I've experienced this because I have a lot of what I do is in the realm of ethics, and so I end up kind of coming off as the ethics police a lot. And people don't like that. They don't like it when you tell them that they're being unethical, or that the thing that they think is going to be really beneficial to humanity is actually not a good idea. People don't receive that well. It's not effective. And so what I would rather see actually is scientists being trained in moral philosophy and in social sciences to understand the dimensions of their science that are not scientific, that are not technological, so that they can themselves be a little bit more aware and receptive to different points of view. Because I think that's the thing is, you know, most people that use it are doing it with the best of intentions, but that doesn't mean that their best intentions are the best way to use it or the right way to use it. And they may be missing an understanding of different kinds of perspectives and different ways of looking at it, like the deaf example I gave you. People, a lot of scientists working in that wouldn't have thought of that unless they had a place or an ability to step outside of themselves and say, well, you know, is there any reason somebody might not want this? And how do I protect that community? So I would say that what I'm doing in that respect is I've created this group where we meet periodically to talk about ethical issues in gene editing, and it's an attempt to do that, to try and, you know, not necessarily criticize the exact application of the scientists in the room, but to just start talking about, you know, what is ethics? What are the ethical considerations that you're seeing, and how should we think about them? And what are the legal questions? And so we bring ethicists, lawyers, social scientists together with a lot of our younger scientists. And the hope is to kind of create a next generation of scientists that will be more alert, more aware, more receptive to different perspectives.
Okay, thank you.
Mm-hmm.
Let's see, we'll try the hand raising thing. Ian, if you can lower your hand. Kathy, your hand was raised. I don't know if that was on accident or if you have a question. If you have a question, now's your time. Going. All right, Esfandiar, your hand is up.
All right, thanks. Thank you so much, Leo, for the great presentation. Very informative. Sorry if you hear a dog background. Let's see. Yeah, I'm definitely intrigued by the whole risk aspect of this. You talked about the technical risk, and it seems like you guys have a good handle on it, or the community has a better handle on that, on the technical risk. It's the social and ethical one that's kind of a scary one, right?
Yeah.
Given what we, the technology usually is way ahead of even society getting the appreciation of what it can do. One good example is what we have or are going through the social networking, like Facebook and like Google. We started using them 10 years, maybe many years ago, and then later we found out how damaging they could be in the society politics. So it's kind of, Ian asked, what could you have done early on? My question is like, and you probably touched upon it a little bit. What are the concrete, are there like an organization beyond IGI, or like are the governing institutes worldwide, nationwide, that are really tackling this? So even though the common people don't understand the impact of this, but there is some frameworks to prevent anything catastrophic happening.
Yeah. So maybe the example I will give you is with the germline editing, the heritable editing. So that kind of potential actually comes on top of a fairly long history of discussion of that concept of being able to engineer children. That kind of global conversation started in the 70s with recombinant DNA, meaning when we first knew that you could add pieces of DNA and move them between cells and between species. So that was already kind of on the minds of scientists and on the minds of the public when we gained that ability. So some discussion was had about, well, how should you use it? What is the risk? Particularly the biosafety risk, meaning kind of contaminating other organisms that you don't want that DNA to go in. There's a lot of regulation put around the biosafety risk in the 70s, starting with, I'm totally forgetting the name of the committee, but there was an entire task force assembled. It's now part of the MSF and the NIH have regulations that you have to follow. There's the RAC, which actually we could talk about that. That's a recombinant advisory committee, which holds scientists as well as ethicists, and you have to, new potentially risky applications have to be evaluated by the RAC for the NIH if you're trying to use government funds for a project. So there was actually a lot put in place that's applicable to genome editing early on. The kind of heritable question came up again with the advent of our ability to do in vitro fertilization and with cloning, with Dolly the sheep. So there was a lot of conversations around that, again, about, how do we make sure that this isn't being used to engineer embryos? And some countries put in place laws specifically about that. So there are countries that have genetic mutation laws that say that you cannot manipulate the human genome for the purposes of reproduction. The US is not one of those countries. So what we did instead was we did a line in the national budget that forbids the use of US funds to fund any kind of scientist work making mutations in the genome. So that's it. And that is still the only regulation that prohibits manipulation of the germline. Germline is cells that can be propagated to future generations. So there's a patchwork of different kind of country regulations that existed already before genome editing came along because of the known risks from cloning and in vitro. I think the difference is that none of those fears came to pass, right? It turned out in vitro fertilization was not horrible, and people, you know, didn't get born with these crazy mutations, and it was fine. I think there's still things that we could discuss about the way it's been used and whether that sometimes crosses the line. But by and large, many of the fears did not come to pass. Mm-hmm. And part of that is, I think, because of technological feasibility. You know, these were like really kind of out there imaginations of, okay, we can make a test tube baby, and then in this other context, we can clone a sheep. But then putting those things together was still so far beyond the realm of technological feasibility that I think it was in some ways easy to grapple with the concept ethically and nationally when it wasn't possible. But now with genome editing, it's possible. Like it is so easy that the first person to do it is actually a biophysicist. He's not the guy that did this in China. He is not trained in medicine. He doesn't have any kind of molecular biology training. He's a biophysicist, and he did this. That's how easy it is. So I think it presented a very different kind of immediacy to the risk that challenged a lot of the regulatory structures that were already in place. And the other challenge is, you know, and this is the challenge in any kind of dual use technology, which is you can make laws and you can make regulatory bodies, but there's always going to be actors that work outside of those. And so what do you do about that? And the general approach is to make it as hard as possible to work outside the law, right? The problem is that we've seen in some cases that's backfired. So for example, with mitochondrial replacement, this is where if you have a mitochondrial disease, some countries have decided that it is okay under certain circumstances to transfer the mitochondria from a donor cell into the embryo. So that's sort of a form of genetic manipulation, but it's a little bit different. You can do that in the UK, for example. It's not allowed in the US, and what that has led to is what would be called like medical tourism. So there's a lot of these really fraudulent clinics that have popped up in, you know, low regulation countries where there's a lot of sketchy stuff happening. And you get families who just really want this, and they will travel to these places that promise them the ability to, you know, have a child that looks like them. And they'll pay for it. And there's really no regulation over that. And so you end up both with risk to that family because it's a poorly regulated procedure, and with risk, you know, to the society at large, because now you have very rich people who are going to get certain kinds of heritable benefits outside of the regulatory process. So I don't know if I'm answering your question, but there are very few, there's international treaties about this. Not every nation has signed on, including the US. There are, there's the WHO, which advises the UN, is doing a whole study on how to regulate human applications, including embryo editing. That will go to the UN and they'll vote on it. But how do you enforce it? There's this is a persistent issue, I guess, in global governance, is that there's really very little that you can do to regulate and enforce.
Thanks. I mean, I appreciate the answer. And I just quick follow up on that. It seems like it's, we talked about maybe rogue individuals or scientists or clinics, but also it seems like could be a rogue nation
yes.
into this, right?
Yeah. So there's a clip actually of Putin talking to some young scientists about germline editing. And he mentions, well, what if you could engineer soldiers to not feel pain? And there's kind of debate about whether he said that as a way to scare them, that it's a bad idea, or whether he said that as like, wouldn't that be great? But you could imagine a place like North Korea or, countries that maybe violate human rights in certain circumstances, feeling okay engineering humans for certain things. I mean, the US government has a program to look at ways to protect against radiation. This is extremely important for things like people undergoing chemotherapy. It's a temporary protection. It's not, a heritable thing. It's not permanent. But that could also be a dual-use technology where you could see that being used by a government like Russia to, justify nuclear attack because they can protect their people. Those are, biosafety dual-use concerns.
Let me go back for a second. That in many ways, because your talk is so good, it really points out the really failures of not only our educational system, but in many ways our culture, that we separate ethics, social systems, and technology. And now, because many of the things you're talking about could have really been foreseen right from the very beginning.
Yeah.
And the thing is that, yes, I understand the tone of ethicists is often off-putting, like we know better than you and all the rest of it. But we can't continue down this path. We can't continue to invent and dump these great, technologies on the world and then later clean up their worst after effects. Something else has to be, and I know the word regulation is dirty, but it almost leaves no alternative unless we start. I mean, the worst unintended, the abuses, misuses, almost could have been seen right from the very beginning. And if we don't learn to get a handle on this and have a better intervention, I'm afraid there's going to be a backlash, what I've called techlash, in a book that I've written. And in many ways, we're asking for it. And it works to our detriment because it puts a cap on technologies that are needed. But you raised it yourself. The fear is designer babies from those who can afford it. So again, let me just stop because I'm going on, that it really reveals the serious flaws, really, in Western civilization. This separation of different fields is untenable. The more that this technology reaches, we can redesign all of human beings. Let me stop. I'm ranting. I don't mean to.
Yeah,
I mean, it is very concerning.
The thing that we struggle with is how to balance those alarm bells with
the benefit,
with tempering backlash, like you're talking about, and tempering expectations. Because yes, on one hand, it is very usable, but on the other hand, there's still a lot of things that are science fiction. So, like I mentioned, the height, right? That is controlled by 100 genes. So there are many things like that where you could imagine, if you have this ability to manipulate the genome, you could do anything you want. Well, we're still fairly constrained by our understanding of the genome. And so how do we manipulate genes to make somebody taller? It turns out that's not so simple from the genome perspective. The technology might be simple, but to use the technology in a way to get the output that you want is actually still, biology questions, basic biology questions. And that's not as straightforward. So there's still sort of this separation between realizing it can be used in certain ways and that it's more immediate risk than it used to be, but also recognizing that in order to do that in a intentional way, there's a lot more that we have to understand about the genome still. There are, so in terms of the dual use stuff, I would say that that's where there's probably the most existing regulation. There's quite a bit at the national level that is constantly doing horizon scanning for dual use technologies. There's approaches to regulate the types of genetic sequences that you can order online, so that you can't, for example, order smallpox sequences. But people have shown ways to get around that, and that raises, continuing. It's kind of like a whack-a-mole. You find a way to plug the hole in one spot, and people find a different one, and so it's just sort of this constant battle of for the biosecurity field to constantly be trying to understand, well, what's the next biosecurity risk that could be presented, and how do we plug that hole? And that tends to be a pretty internationally collaborative field, which is a good thing.
Peter.
Hi, thanks for the presentation. A couple of questions. So early on, there was a kind of summit, right? Like so in the Bay somewhere, right? So Doudna and then also Church's group from Broad and everyone got together to discuss these sorts of things. Were you there? What was it like? Has there been an update of it? Is there a plan for, I mean, this is essentially a kind of self-regulation, in the absence of the speed of regulation, from the government.
Exactly.
So that's question one. Yeah.
Yeah. So I think you're referring specifically to the germline editing question. Yeah. Yeah. So there's the initial meeting was a fairly small group of people. It was Jennifer and then I think 2 bioethicists and a handful of other scientists and lawyers. And it was really just sort of to raise the concern and ask, what do we do next about it? And that meeting was in Napa. That was in 2015, I think, or 2014. And the answer that they came up with at that meeting was, well, we need to have a bigger conversation and we need to bring more people together to talk about this. And so that led to the first international summit, which was on the East Coast. And that's where George Church was, and all the big players in the science field, as well as quite a few science and technology experts, social scientists, and bioethicists and regulatory experts all came together to talk about this and to understand what is the global regulation, what are the concerns, et cetera. And that led to an output from the organizing committee, which was a publication in one of the most regarded journals in the field, calling for essentially a moratorium on using editing in germlines. And it raised a number of issues. Some of them were social issues, but most of them that it raised were technological issues. So, this was still early on when we really didn't understand off-target effects. And I would say even now, the difference between, our comfort level with the off-target risk in adult cells that are non-heritable is that these are not passed on. But if you make a mistake in a heritable cell, that's passed on. So it's a different level of, it might be an infrequent mistake, but it's a very impactful mistake. So there's a lot of attention in that about the technology is not ready. It's too much of a technological risk to try and do this in an embryo. At the meeting, there was some discussion about the social risks of doing it, of creating heritable advantages in certain groups over others. That made a little bit of its way into that report. But as you said, that's a soft governance mechanism. That's, leaders in the field calling on other leaders to not do it. That led to a National Academies consensus report where they created a committee. They did a lot of evaluation and discussion. They put out a report. A number of other national academies around the world did the same thing. Most of them were all reports, which are, we shouldn't undervalue the value of self-governance because it tends to be very agile, whereas hard governance like laws is very slow and it's not agile. So there's a very big role for soft governance, and it appeared to be working very well until the He Jiankui case in China, where, you know, he went against every kind of international norm that had been set, saying, don't do this right now. And it turns out that China is now thinking maybe he broke laws too. So he broke the hard governance and the soft governance. I'm forgetting where I'm going with this, but yeah, I think most of the soft governance efforts have been mostly focused on the technological risk and what would be needed. So for example, the most recent consensus study was a collaboration between the US National Academies of Sciences and the Royal Academy in the UK. And that report, that committee was tasked with saying, if a country were to decide that it was ethically acceptable in a certain case, what would you need to know about the technology to be able to do it safely? So they were really charged with trying to delineate what kind of advances in the science would be needed to make it technically safe if somebody else, so kind of offloading that responsibility to the governments if they decide that it's ethically acceptable. So it's continuing the separation of the technological from the social and the ethical, and that's what I see as a major problem, but it's also a very hard problem to solve. You know, ethics and values and norms are really the realm of governments and not, it's very hard to put those into scientific practice beyond training people to understand it. So
right,
that's
although
really
I do recall from that time that there was so I'm here in Cambridge, so I sometimes, you know, go to these lectures where I run into these folks, and I've been following this for a while, and I recall the something. So Kevin Esveld, who's at MIT, was doing a research study and decided, you know, to focus on an island to release these modified mosquitoes, but at the same time you had a private company, Oxitec, that was saying, no, we have an imperative to go into Africa and test this widely, because the only, you know, or I mean, I guess they were testing it in Florida in some community as well, but like their ultimate goal was to go and, you know, say, you know, what's the alternative? We may as well try it. And, you know, that's a rather difficult tension, right, between the sort of semi-public ethic and the private ethic, you know, the private corporate ethic. And so I'm just wondering, like, you know, where the future of this is, and I guess you're answering that.
Yeah, I think yes, that's right, and especially so the case that you just mentioned is gene drives.
That's right. And
you know, even if it's not a drive, there are certain genetic technologies. So Oxitec is not a drive. Well, it is a drive actually. It's not editing, but it's a drive. But there are other things, like, for example, people have proposed in the coral reefs. So we know that, you know, coral reefs are dying because of water temperature change, because of global warming. But there are some reefs that have fared better than others. And scientists are trying to figure out why. You know, why did this reef survive and this one didn't? And at least in part, there seems to be evidence that some of those that survived had microorganisms living in the reef, in the coral, that had certain genetic traits that allowed them, the microbes, to survive in a warmer climate. And the microbe is actually really important to the survival of the coral. And so there is a discussion about, well, you know, how can we enable the other reefs to survive? And one way to do that would be, let's say we take the bacteria or the microbes from the surviving reef, and we move copies of those into the dying reef. Well, the problem with that is then you have a monoculture. You have one genetic strain of one kind of bacteria all over the world. So if something were to happen that those bacteria were susceptible to, it would all die at once. So the other idea is to figure out which gene is it and move that gene into the natural bacteria in each region. So now you maintain biodiversity, but you're enabling resiliency in coral reefs, which is extremely important for, somebody here was working on flooding. It's huge. It's beyond just kind of the biodiversity, the value of having a reef. There's a lot of human value in protecting shorelines, et cetera. So it's a very important thing to keep from dying. But there's a debate because that's not a drive, but the bacteria are not going to respect national boundaries, right? So if one nation decides we want to do this because we think the risks are outweighed by the benefits, and they embark on that, but their neighboring country says, actually, we think the risks are not outweighed, there's no way to tell the bacteria, well, you're going to have to stay over here because that country doesn't want you. And so this presents a global governance challenge. And I think you're getting at part of the really messy bit of that challenge, which is you have academic actors, individual actors, but you have corporate actors and company actors that have conflicting motivations. And that's compounded by, this ability to cross borders if you're doing environmental release of certain engineered traits. And I just want to be clear, it comes off as very scary and there's all kinds of horrible things that could go wrong. But again, I want to emphasize that when we think about these risks, we also have to think about the risk of not doing it. The risk of not doing it is that we lose all the coral reefs in the world because there's no other solution that anybody has come up with. And so there's a lot of stuff like this that is truly presenting real dilemmas that don't have easy answers.
Right. That leads to the second part of the question, if you will, is that is I'm wondering when I saw the picture of the old hybrid corn breeding and then the modern version, it struck me that the old version of Mendelian whatever messing about is it allows for some period of time before observing the consequences.
Yeah. Yeah.
And CRISPR is the sort of the opposite. You just press a button and you see the consequences. So so the speed to seeing the consequences is tied to another issue, which is the speed of reversibility or the possibility of reversibility. So I wonder if you could just comment on this, observing the consequences and a time to see it, and then also the possibility of reversing consequences,
Yes.
especially if they're made in haste.
Yeah, that's great. That's a great comment. So I would say that this is a case where the concerns about the focusing on technological things is actually really useful, because by implementing the technology, you're creating technological risk and societal risk, but it may actually be implementing technological safety nets that can allow you to mitigate those risks, even if they're social risks, right? So an example is actually with gene drives. So the concern there is that the point of a gene drive is to spread in the population. That is the intent, is spread. But the concern is that what if it spreads where you don't want it to spread? Or what if it turns out that killing all the mosquitoes is actually a really bad idea? What do you do, right? So there are projects actually. So I would say, let me take a step back. I think the gene drive space for genome editing is actually a space that has been very well self-regulated. Pleasantly happy to say. Kevin Esvelt, he mentioned, was early kind of red flag waiver. I think that's another example of a person who was aware enough to be able to draw other people's attention to a potential risk. And what that's led to is something very similar to what happened in the germline space, which is, you know, the experts getting together, talking about the risk, what should we do, should we use it, should we not use it, and putting together kind of self-governing guidelines about, if you're going to do experiments with gene drives, these are the protocols that you need to follow to make sure that it doesn't escape the lab. And those are self-governance, like there's no force of law. And in some cases, funding agencies have adopted those self-governing recommendations into their own funding requirements. And I think this is an interesting way of enforcing self-governance, because most people are not going to be able to do an experiment without a funding source. And so if the funder is able to put in place these requirements, like if you're going to do gene drives, we want you to follow those people's guidelines. All of the largest funders have signed on to a public letter saying that they will uphold those recommendations for funding gene drive experiments. Another way to do it is through publications. So a lot of people have called on journals to enact some of these guidelines, saying that we won't publish your work unless you demonstrated that you followed those guidelines. So that's still soft governance, but it's extremely effective because people want to publish. People need money to do their experiments. And so that's a quick and easy way to implement safeguards. And so that's happened very effectively in the gene drive space. And I would say that the other thing that's happened is part of that kind of red flag waving that Kevin Esvelt did was create a whole subset of the field, which was focused on creating mechanisms of reversing gene drives. So to come up with ways, there's actually a government-funded DARPA program, DARPA is in the defense arm of the government, that is funding multiple projects to reverse gene drives, to come up with ways to undo damage. And they care about this because what if a foreign government came in and did, you know, something like this? So they're funding all kinds of projects. It's called a Safe Genes program, where they're coming up with technological ways to undo, you know, these quick repercussions that you mentioned, to be able to quickly react to it. And one thing, for example, with the mosquitoes that people have talked about is, well, if you're going to release it in the space, maybe you take a population sample of those mosquitoes and you save them, just in case it goes wrong. And if it goes wrong, you know, you inoculate those mosquitoes with like an antidote, basically, and then you re-release them in the environment, and now you have your almost original mosquitoes back. So people are trying to come up with undo buttons for these things.
Leah, let me follow up on Peter's point that one of the frameworks that we use is the following. We look at preventable versus unpreventable crises versus reversible and irreversible. And the thing that obviously raises the real red flags are the unpreventable, irreversible. And that is the thing that I, you know, given your talk, there are all kinds of red flags. I mean, I understand the benefits and the risk of not taking a risk, I understand that. But the thing is that we are venturing more and more into the quadrant of irreversible, unpreventable. And what do we do to get a signal and get a control? I understand about global governance and all the rest of that, but I'm afraid, I'm really, you know, that we have entered mightily with all the technology, not just yours alone, not just the one you're talking about, but we're heading, you know, race storming through the irreversible, unpreventable. And I, you know, more and more, I really worry whether self-governance or self-regulation is up to the task. I realize the dangers of regulation, but I don't see the alternative. And I'm afraid it's, you know, it's just the forces are, and it seems like Europe is ahead of us in terms of regulation. And I wonder whether that's going to become the norm here as well.
It depends on which application you're referring to and who you talk to. So in terms of genome editing in agriculture, there are some people that would say that they're ahead of us because they don't want to see genome editing in agriculture. There are other people that would say they are in the dark ages because they've instituted so much regulation that there cannot be any genome editing in agriculture in Europe at all. And so again, that's a risk of not acting. So I've taken the approach, we've had consultations with the USDA, I've led efforts to respond to proposed regulatory movements at the USDA towards genome editing. And I'd say that we've taken the approach of looking at it kind of in the middle, that there are things about what Secretary Perdue said that are concerning. There are also things about it which are very scientifically accurate. But how do you engender trust and engender an ability to know what's happening and to catch a mistake? And so one of the things that I wrote in our comments to the USDA about their proposed regulation was, if you're not, yes, we agree that there are certain types of editing that shouldn't be regulated as GMOs, but that doesn't mean that they shouldn't be registered or some way of knowing what's happening. Because without any regulation, nobody has any way. There's no place that you can go to know, this company is making this thing. These are the released crops in the environment. These were the edits that were made. And we're still early on enough in the technology that we think it's going to be fine, but without a way of tracking it, we won't know. And then the cow happened, and it's like, that's the example, right? Is we thought that this was going to be fine. Somebody didn't do their work properly, and it wasn't. And so if we hadn't registered that somewhere, even if we didn't put them through the ringer, right? I think the problem with the European regulations is that they are so burdensome and expensive to do that it completely shuts down the entire innovation, regardless of how you use it. But we need something in between where it's protecting, but you're not shutting anything down. And so we proposed a registry of just saying, you are required to register your edit, to say what you're making, to say how you characterized it. And that's it. That way we can do periodic audits of your work. We can see, we can sequence it if we want. If we hear a report of some, unintended consequence, we can know, is it traced back to that thing or not? And the benefit of this is beyond just kind of biocontainment. It's also social benefit because it engenders trust and transparency in the government. And that is one thing that's been lacking with the GMO debate, is people are worried that they don't know what's in their food, they don't know what's happening, it's concerning them. And so if you continue to take away transparency, you're going to create even further backlash that shuts even the good applications down. So that's the approach we have taken. Unfortunately, they didn't agree with us, so they didn't make that. And this is also the problem. Yeah, these are dilemmas. There's not a clear solution, but that is what we've advocated for. And unfortunately, the US government did not agree with us.
Shocking, right?
Shocking.
All right, I think we are nearing the end of our time. If anybody else has a quick question, we can sneak it in. Otherwise, this has been a wonderful presentation and brings up a lot of the concerning issues that we grapple with every day, maybe in different disciplines and domains, but there's a shared anxiety, I think, out there as well. So thank you so much for your presentation and your time today.
Yeah,
Thank you.
Thank you.
Very good. Thank you, Leah.
Yeah.
Thanks.
Yeah.
Bye-bye.
Bye.
Thank you.